Hello and welcome to our today's webinar hosted by the YSA, the Young Scientists Association of the Medical University of Vienna. After our kickoff online event, the YSA Autumn Colloquium on Rare Diseases during COVID-19, which you can still watch on YouTube, it is an honor and a pleasure that our today's prominent expert, Professor Florian Krammer, agreed to join us and give a talk about SARS coronavirus 2 vaccines in development. This is a live event that is recorded, and we would be very happy to bring in your personal questions during the discussion after the talk. Therefore, you can either write in the activated chat function, or you can also click on the hand raise function so we can unmute you and be able to talk to you. My name is Michael Winkliner, and I'm very much looking forward to our discussion. But now I would like to hand over to our today's moderators, Maria Strobel and Ramona Ricke, who will guide you through the webinar.
Very good. Thank you, Michael, for the introduction. My name is Maria, and this is Ramona. Yes, and as Michael said, we're going to lead you through our webinar today with Florian Krammer on the coronavirus vaccines that are being developed at the moment and the current status. And yeah, most importantly, we really want to encourage you to seize the opportunity to ask your questions after his talk, as Michael has mentioned. So this is really a good opportunity to ask your burning questions on the topic. So I would now like to introduce our speaker. Florian Krammer graduated in biotechnology from the University of Natural Resources and Life Sciences in Vienna in 2007, and he also did his PhD there. So during his doctoral studies, under the supervision of Professor Grab, he investigated different kinds of expression systems for recombinant glycoproteins and VLPs, and he focused on influenza virus vaccine design. He completed his PhD in 2010, and he then went to Professor Peter Palese's lab at the Icahn School of Medicine at Mount Sinai in New York, where he started working on a universal influenza vaccine. So actually, very recently, he published very promising and very interesting results concerning this universal influenza vaccine in Nature Medicine. I think you should also read that if you're interested in influenza vaccines. So since 2014, he leads his own research group, and since last year, he is professor of vaccinology at Mount Sinai. And his lab is also part of the NIH-funded Centers for Excellence Influenza Research and Surveillance. So certainly he focuses on influenza, but he has actually already very early during the coronavirus pandemic started to shift his research to the new virus. And which I think is very important also to mention because it's very valuable. His lab is actually providing protocols and reagents for COVID-19 testing and also for performing research on the coronavirus. So alongside approximately 40 publications on SARS-CoV-2 since the beginning of this year, he also published a comprehensive review on vaccine development in Nature, which I think probably many of you have already read. And also important to mention, I think, is that Florian Krammer is also very active on social media. I would say a science communicator, and he shares his opinions and also his expertise, of course, concerning the vaccine development for SARS-CoV-2. So we are very happy that you followed our invitation for the webinar today, Florian, and we are now really very much looking forward to your talk. So you may start sharing your screen.
Thank you very much for this very nice introduction, and thanks for the invitation. Very happy to talk on vaccines and immunity here today. I hope you can all see my screen, and I hope you can see my laser pointer. All right. Okay, so let me get started. I'll start with a little bit of immune responses to SARS coronavirus 2, what they mean, and then how that relates to vaccine development, which vaccines are in development in general, which ones are very far, and what we can expect from them. And so let me just start in the beginning with SARS coronavirus 2 and the antigens that are present there. You're probably also familiar with this schematic of the virus. SARS coronavirus 2 is a typical coronavirus with a fairly large RNA genome on the inside that's protected by a nuclear protein. We have a lipid membrane, which is host cell derived. In that membrane, we have two membrane proteins, the envelope protein and the matrix protein. And then we have the spike protein, which is on the surface. Here on the right side, you see a crystal structure of it. That's actually the protein that's most important for immunity, and it's most important for vaccine development. It's a trimeric surface glycoprotein. It's fairly large, and one of the trimers here is one of the monomers here is shown in dark blue, and the receptor binding domain of that monomer is shown in red. And so this protein is really the protein that the virus uses to attach to our cells. And of course, if you develop antibodies that bind to this protein, specifically the receptor binding domain, the virus has no chance to bind to our cells anymore, which means it's getting neutralized. And so what we know, and basically that has been shown by many studies since the beginning of the year, is that if you get infected, you predominantly make antibody responses against the nuclear protein and the spike protein. The ones against the spike protein, specifically the RBD, can be neutralizing. The ones against the nuclear protein are usually non-neutralizing, mostly because the nuclear protein is not accessible on the inside of the virion. And we have also learned that we get pretty solid T-cell responses against several of the proteins that are in the virion, but also other proteins that are expressed from the genome when the virus is replicating in cells. So there's a lot of additional proteins that are actually not structural proteins. And typically we see a strong CD4 T-cell response and a relatively weak CD8 T-cell response. My lab specializes in antibody responses, and so I'm going to just show you some data that we have about longevity and robustness of these antibody responses. So we started very early on in the end of February, beginning of March, when we started to have the first cases. And very quickly, we developed an assay to measure antibody responses. I mean, that's what my lab is doing for influenza, so it wasn't that difficult. The idea was then to find people who already have antibodies so that we can ask them to donate plasma, and we can use that plasma to treat severe SARS coronavirus 2 cases. And so we started the program at Mount Sinai, and we started to recruit individuals who thought they had a SARS coronavirus infection or who actually had a confirmed infection. And we looked at the antibody responses, and that's what you can see here. This is for the first 1,300 individuals. For the people who had a PCR-confirmed SARS-CoV-2 infection, we saw that 99.5% of them seroconverted. Very few people did not make an antibody response if they had an infection. For the suspected donors, so those are people who were sick in February, maybe in January, in March, people who had contacts with somebody who had SARS-CoV-2 but didn't get tested themselves. We found that only about 40% of them made antibodies, and that can be explained by the other 60% probably not having SARS-CoV-2 infections. We had a severe flu season in New York as well, and so it's likely that these people were infected with other things. And then we continued to do that. Actually, we also invited all of our hospital employees to get tested. Mount Sinai is the largest hospital complex in New York City, so it's quite a few people. And in the end, we tested 80,000 people so far. I'm showing you here data for 70,000, and of these 70,000, 30,000 had antibodies. And our clinical laboratory, to which we transferred our assay from my research lab, measures these titers in several steps. So you can have a 1:80 or 1:60 titer, which are considered low titers, and about 7% of individuals had that type of immune response. We have a 1:320 titer, which is about 20% of individuals. That's considered an intermediate titer. And then we have 1:960 or 1:2880 or higher, and those are considered high titers. And about 70% of the individuals we tested had these high titers if they were positive. And here on the right side, I'm just showing you samples that were run per day over the last few months. And the gray bars here show you number of positive samples. And the blue line is basically the percentage of of individuals that had on each given day a titer of 1 to 320 or higher, and that oscillates around 90%. The only message here is the antibody response that you make against SARS coronavirus 2 when you get infected, even if it's a mild infection, is relatively robust. It looks like a normal antibody response. That wasn't clear in the beginning, and there were a few papers that suggested that the antibody response was really being suboptimal, but we and others showed that that doesn't seem to be the case. With these ELISAs that we developed, we measure binding antibodies, but we don't measure functionality. And so we were interested to see if these antibodies now can neutralize the virus. That was our working hypothesis, because we use the spike protein here as substrate for the assays. And in fact, when we did a correlation analysis for a subset of individuals, when we looked at ELISA titers versus neutralization activity, which is shown here on the y-axis, we see a very nice correlation. And this is actually neutralization with the authentic SARS coronavirus 2. So this is not pseudotype particles, this is actual virus neutralization that's done in biosafety level 3. The other question was, how long-lived are these antibody responses? And I think you can remember there was a paper back in, I think, April, saying that the antibodies go away after 4 to 8 weeks. And so we wanted to investigate that too. We enrolled about 120 people in the donor program into a longitudinal study. We bled them on day 30, on day 82, and then 5 months out. And in A here, you can see the titers that they developed. So basically, initially, they had a GMT, geometric mean titer of 764. And from day 30 to 82, there was very little change here. And then it started to drop off a little bit. But if you look at the y-axis at the scale here, this is not a very drastic drop-off. So this is something that might not even be biologically relevant. And we also stratified that by initial titers, and we saw that people who have initially a very high titer have more of a drop than people who have initially a low titer of 1 to 320, 1 to 60, or 1 to 80. There we actually saw a little bit delayed kinetics, where there was usually already still an increase from day 30 to day 82, and then a decrease from day 82 out to 5 months. For the same individuals, we have now 7 months data as well, and it's basically a flat line. It doesn't drop any further, and that makes a lot of sense from a B cell point of view. And we also looked at neutralization over time. This is shown here on the left side in G. You have quite a variability of a variety of neutralization titers initially, and there is certainly a drop from here a GMT of 176 to 75, and then to 52. But as with the ELISA titers, this neutralization activity doesn't really. And this has now also been shown by several other labs. And if we at 5 months out do a correlation analysis, again, we see that there is still a correlation, meaning that the epitope specificity doesn't shift in a way that now would prefer or favor non-neutralizing antibodies. All right, so what does all of this mean? Does this mean that we are getting protected, that we are protected if we got infected? And to start with that question, I wanted to show you some data from Dan Baruch's lab in non-human primates, in rhesus macaques, which are a pretty good model for mild, I would say, SARS coronavirus 2 infections. Those papers were very nice. They were published in Science back-to-back this spring. Here on the left side, you see infection-reinfection experiment. So basically, they infected these rhesus macaques with SARS coronavirus 2. This here is RNA copies in the lung. So it's basically a substitute for viral titers. And you see the animals get infected, and then the virus slowly clears, and most of the virus is gone by day 21. So the animals clear the virus, they don't die, they actually recover. And then a few weeks later, they try to re-challenge them. And you can see that although there's a blip here on day 1, which is probably input virus, the animals are protected, so you can't reinfect them. And I know that there is some reports of reinfections that seem to be true, but that seems to be a rare event. It looks like humans behave like these non-human primates in terms of SARS-CoV-2 infections. And in their second paper, they tested a DNA vaccine, which was an experimental vaccine. It wasn't meant to go into vaccine development, and it gave a wide variety of neutralizing antibody titers. And then when they challenged the animals, they again looked at virus in the lung. And if you have this wide variety of neutralizing titers, you can try to correlate the titers with protection of the lung. And that's actually what we see here. There's actually a very strong correlation between lower titers in the lung and higher neutralization titers. So what they managed was to define a correlate of protection for SARS-CoV-2 in these animals, in these rhesus macaques. And the titer is actually not that high. This is a 1 to 100 neutralization titer. A lot of people who recover from SARS-CoV-2 easily reach that. So that was good news. And I just wanted to stress that what we call correlate of protection, meaning an immune readout that tells you if you're protected or not, is something that is very common. We have that for many different vaccines. We have that for many different virus infections. Here's a list that I stole from Stanley Plotkin. And for example, you have this for hepatitis A and hepatitis B. You have this for influenza. This is the famous HI titer of 1 to 40. If you have that, your risk of getting flu is reduced by 50%. We have this for measles and so on and so forth. So it would be very nice to have that for SARS-CoV-2 as well, because that would facilitate vaccine development, and it would be easy to tell people if they, for example, need a booster dose, right? If they're above this number, they know they're protected. If they're below, they are susceptible and they need to be immunized. And we got a little bit of a glimpse of a correlate of protection in humans as well. And this is from a study that was done by the Graninger lab in Seattle. I really liked that study. They basically recruited 122 crew members of a fishing boat that went out to the Pacific. They bled everybody. They determined neutralizing antibody titers in serum, and they found that 3 people of these 122 had neutralizing antibodies. And then the ship left, and as you know from the cruise ships in the beginning of the year, if you get an infection on one of those ships, it's like it burns through like a wildfire, because you have very close quarters, you have a lot of interactions, and people get easily infected. So they ended up with an attack rate of almost 83%. But the 3 people who had neutralizing antibodies did not get infected. And this is a small number, but it's statistically significant. And that was really the first idea that we got about neutralizing antibodies being a correlate of protection. And there was also a recent paper, and this is not peer-reviewed yet, this is still a preprint, by a British group in healthcare workers, where they followed healthcare workers and basically sampled them all the time to look if they get infected, and they also determined their antibody titers. And what the study found is that if you have antibodies to SARS-CoV-2, your risk to get an infection, even an asymptomatic infection, is much, much, much lower. They show this here on this graph on the right side. It's a little bit of a weird way of showing that. Basically, the higher the antibody on the right side are the higher antibody titers. This dashed line here is basically the cutoff. If you're to the left of the dashed line, you're antibody negative. If you're to the right, you're antibody positive. And then on the y-axis, we basically have the risk of getting infected. So there's more and more data suggesting that if you have antibodies, you might be protected. And there will be a lot of studies coming out in the next few months, including a large study from my laboratory. We also have similar findings already. So just to conclude on this antibody part, humans induce solid antibody responses to SARS-CoV-2 even after mild infection, and most of the people in our cohort were actually mild infections. These antibodies that bind to the spike protein correlate with neutralization, which makes a lot of sense, and is actually what we see with other coronaviruses too. And the antibody response really looks normal. We see an initial strong increase, which is driven by plasmablasts. Plasmablasts are types of B cells that expand quickly, make a lot of antibody, and then die after a few days. But the antibody that they pumped out has a half-life of about 21 days, so that stays around for quite some time, even if those cells are gone. But of course, at some point, this starts to drop off. And then it basically levels off and stabilizes, and that stable level of antibodies is actually not produced by plasmablasts, but by long-lived plasma cells in the bone marrow. And those cells can live for a very long time and can actually produce antibody partially lifelong. So this looks like a very, very normal antibody response. The only question that's left on individual level, if you add up at this stabilized antibody titer, is that titer above or below the protective threshold. And again, we need to figure out what that threshold is. But let me switch gears now to vaccine development. So I wanted to start out with just explaining how vaccine development usually works, how long it takes, and then we'll contrast that with what happened this year. And so typically vaccines are developed at a very glacial pace. I started in 2011 to work on a universal flu vaccine that went into phase 1 trials in 2017. Now we got the result. So it's many, many years usually. Typically, you start in an academic laboratory with a design and exploratory preclinical phase, meaning you play around with different antigens, different vaccine platforms, you vaccinate mice and you see if they are protected. That might go on for a long time, until the point where you think you have something that's very promising. When you have that and you want to develop this further, you need to look for a commercial partner. Because typically an academic lab can't really develop a vaccine. You need a company for that. So you look for a commercial partner. If you're lucky, you find one who is interested, and then more formal work starts, right? You need a process to make the vaccine in the right quality, in GMP quality. Otherwise, you can't go into humans. You need to do more preclinical studies, toxicology studies. And once you have all of that, which can take 2 to 4 years, you submit an investigational new drug application if you're in the United States. That's a document that goes to the FDA, has all the data. And then you start a phase 1 trial. And so a phase 1 is basically a trial in usually less than 100 people that evaluates safety and a little bit of immunogenicity. So safety is really the focus there. And that takes 1 to 2 years. Then you go into phase 2 if phase 1 looks good. If you see a safety signal, if something happens here, this doesn't go forward to phase 2. So then you go to phase 2, which is usually a few hundred people. You look for immunogenicity, safety again. You try to test different doses, different intervals between vaccinations, and so on and so forth. Takes another 2 years. And then when you have the results from phase 2, you have to really sit down and think and analyze if you want to move forward. So if the vaccine looks promising, if you still think you have a market for the vaccine and you think that it will not fail in phase 3, and you get all the funding that you need to move it forward to a $200–300 million phase 3 trial, then you move forward. Before you start phase 3, you're going to determine how success looks. You have to determine the criteria for success. And then you perform the phase 3 trial. And I'll explain in a few minutes how that works. But basically, in the end, your results basically need to meet your success criteria. Otherwise, it's not getting licensed. But anyways, you do the phase 3, which is usually 2 to 3 years. If everything looks good, you can submit the biologics license application. And then the FDA, or in Europe, the EMA looks at it. And they might decide to license your vaccine if everything looks good. That's still quite a process usually, not because it couldn't be done quicker, but because there's many licenses license applications that are around, and there's a lot of work, and usually people are understaffed, and it's quite a process in terms of administrative review. And only after your vaccine is getting licensed, you start large-scale production and distribution. you don't want to do that earlier, because what if it doesn't get licensed? Then you kind of made millions of doses and you have to throw them out. All right, so what happened for SARS-CoV-2? Design and exploratory preclinical phase were skipped. There was enough work on SARS coronavirus 1 and MERS to know what to focus on, and that was the spike protein and how to modify the spike protein to make it express well and very immunogenic. The sequence of the new virus was released on January 10th, and I believe on January 11th, most of the vaccine companies knew how their construct would look like. So there was no fussing around. Specifically for things like RNA vaccines or vectored vaccines, they just took processes that were already established for other vaccine candidates. For those technologies, they just switched the sequence. The process doesn't change. And so there was not much work put in there either. For some other companies like Novavax, for example, it makes a recombinant protein vaccine that took a little bit longer, and that might be the reason why they are now a little bit behind. But basically, a lot of this already existed and could be skipped. INDs were submitted. Moderna was the first company to go into a phase 1 trial on March 16th. And they did a phase 1 with an adaptive design. And once the phase 1 interim results looked good, meaning the vaccine looked safe, there was immunogenicity, they increased the number of people that were enrolled and added additional groups. And this became a phase 2 trial. And then the phase 2 trial had an interim analysis that looked at safety and immunogenicity. And of course, in the meantime, a lot of non-human primate studies were going on. And after this interim analysis, phase 3 was started. Phase 3 for a lot of the vaccines that we see right now started in July. So they started to enroll and vaccinate people already in July. And at the same time, production started, completely at risk. And so you might ask the question, how was that possible? And the reason is very easy. There was no financial risk here. There was basically endless funding to do this. The resources were put in, and this was given high priority. There was no competition for other resources, and there were literally billions of dollars to do this, even to start production at risk. And so that's why this was done. Money actually made that possible. And then the licensing procedure is a little bit different too. Sorry. It depends now on the country, but in the US, the phase 3 in terms of efficacy data was finished, and emergency use authorization application was filed. So that's not the final application. The Moderna and Pfizer, for example, will have to submit a final one after 2 to 3 years. But that already has a lot of the data that's needed for licensing these vaccines, including most of the safety data and short-term efficacy data. And then the authorities think about that data, and they put that into the context of what's going on in terms of the pandemic, right? You also take into account that thousands of people die every day, and you take into account that a lot of people are getting sick, that the ICUs are full. And so that's why they allow for certain things not to be there. For example, a 2-year efficacy doesn't have to be there. It's fine if the efficacy is just looked at for a few months. But again, the data will need to get submitted later on too. Other countries like Canada actually gave a full license. So it depends a little bit on the country that does the licensing. And so that's the reason why this why it was possible to contract that to approximately 10 to 12 months. A little bit about the candidates. Here in the middle, I'm showing you the model of the virus again and the spike protein. And what we see now is actually the biggest experiment in terms of comparing vaccine platforms with the same antigen that has ever been performed. It's actually very nice, and we'll learn a lot for other vaccines when all of this is over. But basically, there's many different vaccine platforms that are used. One of the first ones that was developed for SARS-CoV-2 was inactivated vaccines. These vaccines are mostly developed in China and in India, but I think Valneva in Vienna is now also starting with one. You basically take the virus from a patient, you grow it in viro cells, then you inactivate it, usually with beta-propiolactone, you concentrate it, and you use that as vaccine. You can also use an attenuated virus, and that's something that has been classically done for a lot of vaccines. Measles vaccine, for example, is an attenuated vaccine, and there's two ways to do that. You can either isolate the virus and then passage it on animal cells, and the virus will slowly adapt, mutate, and like those animal cells now better than human cells. And when you use it in humans, it doesn't really replicate that well anymore, doesn't cause disease, but still gives you an immune response. The other possibility to make live attenuated vaccines is to just manipulate the genome and take away part of an open reading frame, for example. And that might also lead to a virus that still replicates a little, gives you an immune response, but doesn't cause disease. Candidates for SARS-CoV-2 that are based on that technology are way behind for many reasons, because this adaptation process takes time, because the genome is not so easy to manipulate. So they are still in the preclinical stage. Then we have virus vectors, and some of them are really, really far. And you can have two types of viral vectors. You can have viral vectors that still replicate. Those are usually vaccine viruses. For example, you can use a measles vaccine, and you can add the information for the spike of SARS-CoV-2 to the measles genome. And then when you get vaccinated with the modified measles virus, the virus infects cells, and they also express the spike protein of SARS-CoV-2. So that's a technology that can be used. And then you can have non-replication competent vectors. And in this case, you also take a virus, adenoviruses, for example, are used very often. You take away parts of the genome of that virus, so the virus cannot replicate itself anymore. It still can go into your cells and deliver information, but it can't replicate anymore. And you replace that with the information for the SARS-CoV-2 spike. And this is actually an approach that was taken by AstraZeneca with a chimp adenovirus, by Janssen with YSA’21, and also by Russian and Chinese companies. Then you can make recombinant protein vaccines, either based on just the RPD, so the little red part here, or the full-length spike, or you can make virus-like particles. And one candidate based on the full-length spike is actually very far in clinical trials already. Phase 3 with that candidate from Novavax was started a few months ago in the UK and in South Africa. That looks very promising. Then you have DNA vaccines, which were started in phase 1 trials quickly, but then didn't advance much. And then we have our RNA vaccines, which are now widely discussed. And so for these RNA vaccines, just to explain that a little bit, you take the coding information for SARS-CoV-2 spike, you in vitro transcribe that, you make messenger RNA, and that messenger RNA is then packaged into lipid nanoparticles. And those are then injected intramuscularly, and the lipid nanoparticles are taken up by cells at the injection site, mostly muscle cells, and those cells start to use the mRNA that was provided, and they start to make spike protein and display it on their surface. And you get an immune response against that. It's actually pretty simple, but it's a pretty new, relatively new technology. All right, this is a slide that just shows how many candidates there are in development. We have our clinical phases here at the bottom. Preclinical just means animal testing, then phase 1, phase 2, and then phase 3. And vaccines that are licensed. And so this is actually outdated. This is, I think, 2 months old, but at that point, there were 140 serious vaccine candidates in preclinical trials. There were many in phase 1 and phase 2, and there's now, I think, 11 that are in phase 3 trials. And at that point, there were 2 that were licensed, but there's an asterisk there because they were not licensed the regular way. One was the vaccine, the Russian vaccine that was made by Gamaleya Institute, and that was licensed after a phase 2 trial with about 70 individuals. So it didn't actually go through through phase 3 trials. And the second one is a vaccine candidate that's also based on adenovirus that was produced in China by CanSino. And that one was licensed for use in the Chinese military. And by now, there's 2 more licensed vaccines, and I will get to that. One is the Pfizer vaccine, and the second one is a vaccine candidate that is produced by Sinovac. Okay, so for many of these vaccine candidates that are in phase 3, there's data in non-human primates. This is a lot of text here. We're not going to go through this. I'm just going to tell you what the main finding is. So with the non-human primates, it's nice because you can vaccinate them and then you can infect them and then you can see if they're protected. You can do that relatively quickly. And so, long story short, all of these vaccine candidates, and we have here inactivated vaccines from Chinese manufacturers, the AstraZeneca vector vaccine, the Janssen vector vaccine, the Moderna RNA vaccine, and the Novavax recombinant protein vaccine. All of these vaccines protected these non-human primates from getting sick. They protected basically the lung. When you vaccinated these animals, there was no virus replication in the lung. The other finding here was that in most cases, actually in almost all cases, the virus was still able to replicate in the upper respiratory tract. To a certain extent, usually less than in animals that had not been given the vaccine, and usually for a shorter period of time. But in most of these animals, even if they were vaccinated, you still found virus in the throat, in the nose, and so on and so forth. The reason for that is likely that all these vaccines are given intramuscularly, and that's not ideal for inducing a mucosal immune response, which would really protect these parts of the upper respiratory tract. Keep that in mind. We'll get back to that. And then we have also data from phase 1-2 trials for many of these vaccines. Again, I'm not going to walk you through this. Just going to explain a few things. The inactivated vaccine, again, from Chinese manufacturers, worked relatively well. If you vaccinate 2 times or even 3 times with them, you get okay neutralizing antibody titers. The CanSino virus vector, which is based on Ad5, didn't do that well. That induced very low neutralizing antibody titers. But they think it's a success and it's moving forward, and it does induce good T-cell responses. The AstraZeneca vaccine, which is also a viral vector, induced, even after 1 shot, okay neutralizing antibody responses that increased a little bit after the 2nd shot. Then we have our RNA vaccines, and I put the doses that moved then forward into phase 3 in bold and larger here. So for Moderna, it was the 100 microgram dose. For Pfizer, it was the 30 microgram dose. And Pfizer actually did parallel development of several candidates and then chose BNT162b2 as the one that they moved forward. The interesting part here is if you give these vaccines the first time, you get very low neutralizing antibody titers. Usually you can detect them, but they are low. If you give the vaccine the second time, you get a pretty good antibody response. And then we have Novavax. Again, this is a recombinant protein vaccine that is adjuvanted with a saponin-based adjuvant, and they give that twice too, and they get sky-high neutralization titers. So this is basically 10 times higher than what we see with the RNA vaccines. There might be assay variation that is partially causing this. It's very hard sometimes to compare even neutralization assays from one lab to another. But these are outstanding titers. So I have high hopes for this game. And so to sum this up, we have Moderna and Pfizer, which are RNA vaccines, and they are in phase 3 trials. We have this green check here, and this is because both of these companies reported final efficacy data for phase 3. So in terms of efficacy, they're done. AstraZeneca reported interim results for efficacy. I'll show you that data in a few minutes. Janssen has a viral vector vaccine based on Ad26, and that trial is still running. The Novavax trial, which is Novavax vaccine, which is based on recombinant spike, is running in South Africa and in the UK. They were supposed to start a phase 3 study also in the US, but they got postponed. And I hope there is no issues in terms of manufacturing, because this spike protein is not so easy to make. We have the Gamaleya vector vaccine, which is the Russian vaccine. We have interim data for that, efficacy data for that too. We have the 3 inactivated vaccines that are produced by Sinovac and Sinopharm. And for one of the candidates, we have interim data. I'll show you that too. Then we have CanSino, which is also an adenovirus vector vaccine from China. These 3 here are shown in red, basically just because they're different in terms of where they are produced and developed. And it's very unlikely that these will be licensed in the European Union or in the United States. That doesn't mean that they are not as good as the other ones or that there is a problem. But there's a lot of incompatibility when you look at the regulatory agencies. And so it's very likely that these vaccines will be used to satisfy the global demand, but it's unlikely that they show up in Europe. And then we have another candidate, Epivac Corona. This was also licensed in Russia, but I couldn't find any information on this vaccine. Supposedly it went through phase 2 and had good data. But again, there's no publicly available information. So just a little bit about how these phase 3 trials work and what we do there. Typically, you have 2 groups. 50% of individuals get a placebo. That's the control group. That's usually a saline injection, for example, but could also be a vaccine that's unrelated and already licensed. And 50% get the actual vaccine that you wanted. And those studies are double-blinded, meaning that the person who gets the vaccine doesn't know what if it's in the placebo group or the vaccine group, and the healthcare worker who administers the vaccine doesn't know either. And that's to remove any biases that could be there. And then time goes on, and COVID-19 cases are reported in both groups, and all the data is actually not going to the company that develops the vaccine, but to an independent data monitoring committee. And that committee looks over the safety, and it looks over the other data that comes in. And when you say, for example, initially when you start your phase 3, okay, after 60 cases, we want to have an interim analysis, the IDMC is going to tell the company when there are 60 cases, and they will provide them with the analysis. And that's just to make sure that there's nobody who's messing with the data. Okay, so I wanted to walk you through the results that we have so far. Let's start with Pfizer. We had about 43,000 individuals in their phase 3 study, which, as I said, started in July. There were 170 COVID-19 cases, 162 in the placebo group, and 9 of those severe, and 8 in the vaccine group, and 1 of those severe. They got an efficacy of 95% when they started to measure 7 days post dose 2. And that's really good. That's where you want to be with a viral vaccine. There are some viral vaccines like measles that are even better, but this is actually really, really good. Flu vaccines have an efficacy usually of 75%. So this is way better than the flu vaccine, for example. Also be careful, because there's a difference between efficacy and effectiveness for flu, and the effectiveness is usually way lower for the flu vaccines too, but 95% is really good. They had a group in a large number of people in their phase 3 that were 65 to 85 years old. And that was important because, as you know, those are the people who are at highest risk. And they had enough cases that they could also calculate an efficacy in that group and for that group only. And that was 94%. And this is great because that means that in the high-risk group, the vaccine actually works. The people who need it most, it still works in that group. They had no significant safety concerns. And the vaccine now received different degrees of approval in Bahrain, in the UK, in Mexico, in Canada, in Saudi Arabia, and last week also in the US. The meeting where the EMA the European Medicines Agency will decide and discuss licensure in Europe is scheduled for December 29th. And Pfizer said they expect to produce 50 million doses already in 2020. So those are getting distributed right now in those countries, and 1.3 million doses for 2021. And now the vaccine also got a name. It's called Dosinameran. I still have to get used to that. I have no clue what it means. And this is just to show you a little bit of the data. This is from the documents that Pfizer submitted to the FDA, which you can actually download from the FDA's homepage. The FDA did this very transparent. There's always these VRBPAC meetings. VRBPAC is a group that advises the FDA, and there's day-long meetings which you can actually follow on YouTube if you want to. And as part of that, they also supply all the data. And so here's what they saw in the trial. There's two lines. The red line is the placebo control group, and you see there's a steady increase of cases of SARS-CoV-2. And then you have the blue group, and the blue group is the vaccine group. And so remember, they got a shot on day 0 and on day 21. And what you see approximately 10 days after the first shot, this curve, the blue curve, deviates from the red curve. And this is basically when your first immune response kicks in. And it seems that that already provides protection. Of course, there were a few cases along the line in the vaccinated group, but that's normal. But I found this impressive. When I saw this, I was very happy. And this is actually how a good vaccine in a phase 3 trial looks like. Okay, just to go to the competitor of Pfizer, and that's Moderna. The Moderna vaccine is also an RNA vaccine. It's very, very similar, almost identical to the Pfizer vaccine. They had 30,000 individuals in their study. They recorded 196 COVID cases, 185 in the placebo group, 30 of those severe cases, 1 death, and 11 in the vaccinated group, no severe cases. They calculated a 94.1% efficacy, again, no significant safety concerns, and Moderna filed an EUA with the FDA, and that meeting is going to take place on Thursday. The meeting with the EMA is going to take place on January 12th. And Moderna also expects to provide 20 million doses still this year, and then between 500 million and 1 billion doses next year globally. And then we have interim results for some other candidates. AstraZeneca, which again is based on chimpanzee adenovirus, started 2 studies on which they reported, one in Brazil, which is a fairly large study where they reported 62% efficacy, and a very small study in the UK where they reported 90% efficacy. They, in their press release, said that 131 COVID-19 cases, but didn't tell us in which of these groups. And then it turned out that the UK group for the first dose got only half dose, which is something shouldn't happen in a phase 3 trial. That sends a very bad message. The only thing we can conclude here right now is that the vaccine works, but we don't know how well it works. They will need to present more data and consistent data so that we can basically figure out how well it works compared to the other vaccines. Sinovac, which is a Chinese company that makes an inactivated vaccine, did a trial with 31,000 individuals. They recently reported 86% efficacy and got a license in the United Arab Emirates. And that's also great because this is a vaccine that, as compared to the RNA vaccines, which have to be stored frozen, only has to be stored at 2 to 8 degrees Celsius. And so the companies that make this vaccine also have a lot of production capacity that will help to satisfy the international market. And then we have the Russian vaccine candidate, which they called Sputnik 5, which is a sequential vaccination with Ad5 and Ad26 vectors, and they have 95% efficacy that they reported. All right, so a couple of things in the end. The first thing that I wanted to touch on is reactogenicity. These vaccines are pretty reactogenic, specifically the RNA vaccines and the adenovirus vaccines. What's reactogenicity? Reactogenicity is basically symptoms that you might have right after vaccination: injection site pain, headache, fatigue, elevated temperature, muscle aches, basically mild flu-like symptoms. And they are driven by an innate immune response, by secretion of interferon, your body basically saying, hey, there's something in my body that's not supposed to be there. Let's react to that. This is unpleasant, but it's not dangerous. And some of you might have that when you get a flu shot, when you get other shots. And this is usually very transient. It goes away after 24 to 48 hours. And as I said, we see that mostly in the adenovirus vaccines and the mRNA vaccines, but less in recombinant protein vaccines and inactivated vaccines. And here on the right side, you basically see this reactogenicity for vaccines in the Moderna phase 2 trial. Look at the 100 microgram dose here, which is the one that moved forward. What we see is that after the first shot, you basically have almost or have very few of these symptoms and at the low rate, except for injection site pain, which is very common. But if you get the second shot, actually a very high proportion of people experience that. And that's something to keep in mind because this is not as tolerable as, let's say, a flu shot. A lot of people will have these symptoms. And you have to keep that in mind when you get the vaccine. This is not something dangerous. This is most likely going to happen when you get the vaccine the second time. And it basically is a sign that your immune system is responding. Unpleasant, but not dangerous. And then there's another story, and that is also something important to bring up. So the UK started to vaccinate last week, and they reported severe allergic reaction in 2 individuals that got vaccinated. And so what we need to know here is that there are some people that have severe allergic reactions to vaccines, to all kinds of vaccines, and they were actually excluded from the phase 3. But regular allergic individuals were not excluded. So there's all kinds of people with food allergies, with allergies against insect bites, with allergies against kind of, I don't know, pollen, for example, that have been in the study at a similar rate as they exist in the general population. And there were no issues with that at all. The 2 individuals that had these allergic reactions in the UK had a history of severe allergic reactions. They have been, of course, treated. They had an EpiPen with them, and they are fine. But the UK regulatory advice was now updated and says that individuals with a history of severe allergic reactions should not get vaccinated. This is what happened in the UK. The FDA, for example, asked that this is looked at, but didn't exclude anybody with allergies from getting vaccinated. So we'll see in the next couple of days how this develops. Another point that I wanted to make, and that's kind of alleviated by what we saw from Pfizer, is that usually older people don't react to vaccines or respond to vaccines as good as younger people. And that can be an issue because, of course, older people have a way higher risk for severe COVID-19. So just look at the blue bars here, because that's the candidate that moved forward into phase 3. Here we have the neutralizing antibody titers in the 18 to 55-year group. And on the right side, same vaccine, but 65 to 85 years of age. And you do see that older people make lower antibody responses. And this was also seen for the inactivated vaccines and the vectored vaccines. And for Pfizer, it seems that this is not an issue, but this is certainly something we need to keep an eye on for the other vaccine candidates as well. We need data for all vaccine candidates of how well they work in older people. And then another point, and I touched on that already with the non-human primate studies, is protection from disease versus protection from infection. And that's an important point too. So if you get a natural infection with a respiratory virus, you get a mucosal immune response and you get a systemic immune response. The systemic immune response is mostly IgG1. And the systemic immune response protects your lung, and the mucosal immune response protects your upper respiratory tract. Now, if you get a vaccine, you get basically only or mostly these IgG responses, also some IgA, but that's serum IgA, that's not mucosal IgA. And that might protect you very well from disease, but it might leave some of the upper respiratory tract vulnerable to infection. And again, this was seen in the non-human primates, and this is something that we don't know yet if it's going to happen in humans as well. The phase 3 trials had as readout symptomatic disease. So not everybody was swabbed all the time. People only got swabbed when they had symptoms. And so we only know if the vaccines protect from symptomatic disease, and that's what the efficacy tells you. It's not efficacy against infection, and there might be a difference. And it might be the case that some people who got vaccinated can still get infected, can still shed virus, and might still be able to infect other people. However, it's very likely that vaccinated individuals, if they get asymptomatically infected, shed virus for a shorter period of time and shed less virus. So the risk might be reduced, but that's something to keep in mind. And there is actually a way around that. You can use vaccines mucosally, and there's some SARS-CoV-2 vaccines in development that might be given mucosally, and that would also result in a mucosal immune response and then most likely sterilizing immunity. We'll see if that comes as a second or third generation vaccine. Another big question is longevity of protection. Right now, we don't know how long these vaccines will protect us. The efficacy that was measured was short-term, but there's no reason to believe that the protection will be gone within a few months. This is usually not how the immune system works. And there's first data now from Moderna, basically over 4 months, and what they see is, depending on actually across the age groups, the antibody titers go up and they stay up. There's some waning, again, as expected, and we see that with natural infection too. You see that here in the neutralizing antibody titers, but not to a level that would be concerning. And it's very likely that if you follow them further out, that just stabilizes. So we'll see how long these vaccines protect. My guess is for a few years, but I also think that at some point you might need a booster dose, which is not a big deal. We have that for the TBE vaccine. We have that for tetanus. You need to get booster doses for many vaccines. So that's not a another few issues to keep in mind. One is large-scale production. Some of the frontrunners in this vaccine race have never produced a licensed vaccine, including Moderna and Novavax. So when they have to scale up to billions of or millions and billions of doses, they might run into issues with supply chains, for example. And then there's some technologies that have never been used at that scale. And the same thing might happen. If some reagent that you need for vaccine production is not available anymore at that scale, you can't make the vaccine anymore, right? And that's something to look out for. Distribution can be challenging, specifically for the RNA vaccines, which have to be stored at very low temperature. Administration can be an issue. First, you have to figure out who gets vaccinated first, and I think most countries got their act together now and made plans how they want to do that. And then the other question is, how do you vaccinate, right? The UK started with central vaccination events at hospitals. I think today they started to have vaccination also in regular doctors' offices. But that also needs to be figured out. And then the last point is really immunity. While the Pfizer vaccine already shows efficacy after the first shot, until you're really well protected, it takes time. You need to get the first shot, you need to get the second shot, and during that time you might still be vulnerable, and that's kind of something that needs to be factored in too. If Austria starts to vaccinate people in January, those people are likely not well protected until the end of February. So they have to still be careful, and you still have to take care of them. And then, as I mentioned, it's not clear if booster doses are needed. And so with that, I'd like to conclude. We have several candidates that induce strong neutralizing antibody responses in non-human primates and in humans. The protection from lower respiratory tract, meaning disease, in non-human primates is really solid. Protection from upper respiratory tract infection is often only partial. And none of the vaccines currently far in development induce a mucosal immune response. The Pfizer and Moderna phase 3 results look really excellent, so you couldn't wish for anything better. And the Pfizer vaccine is already licensed in several countries. And I'm going to finish that with acknowledging a lot of people, my laboratory at Mount Sinai, and also a lot of collaborators who helped us to get things up and running in the beginning of the year. And I just wanted to mention, I'm currently looking for postdocs, and I'm also looking for students. So if somebody's interested, just shoot me an email. I think the email address is here. And with that, I'm happy to take any questions.
So thank you for the very enlightening talk, Professor Krammer. I think we will go right away to the questions because there are quite a lot piling up, and we will say that we are sorry if we don't manage to go through all the questions because they're really quite a lot. So I will start right away with the first one, which is which cells produce and secrete the full-length spike protein, and how do B cells come into contact with this unprocessed spike protein?
So it's produced mostly locally, right? So the majority, it depends on how these lipid nanoparticles are, the composition of the lipid nanoparticles, but they basically stay locally in the tissue, and it's mostly muscle cells that that produce that, right? And I don't think it's too well studied how the B cells then interact with that. You would of course have antigen presenting cells that carry some of that that spike protein from these cells into germinal centers. Some of these cells that make the spike protein might also basically die, and fragments of that might be carried away, and there might be some local interactions too. So that's not 100% clear.
Okay, next question. Your data regarding the duration of the antibodies against SARS-CoV-2 is very encouraging. And do you expect similar effects for the vaccination, or do you think it will be necessary to perform more than 2 vaccinations?
Yeah, I think that the antibody titers will not just magically disappear. I think they will stabilize and just stay there. But over time, they might slowly decline. And as I said, my best speculation right now is that you need a booster dose every few years, maybe every 3, 4, 5 years, something like that.
Maybe I can go on with the next question, which I think is also very interesting. Would you recommend getting vaccinated? So for people who have already been previously infected with corona?
Yes and no. I don't think they should be a priority. We know that there's no issue, no safety issue if they get vaccinated, because in these phase 3 trials, there were actually people who already had antibodies, right? And they were enrolled. It's a minority, but they're there. And they have sky-high antibody titers now. But I think if you already got infected, you are most likely protected, and you're most likely protected specifically from severe disease. So I think it's not really most efficient to vaccinate those people right away. Sorry. But basically, if you look at the bigger picture, it might be very tough to everybody if they already have antibodies before you administer the vaccine. And so the decision in the US at least was that for now they vaccinate healthcare workers and nursing home residents, and they're not going to discriminate between people who already had the infection or not. They're just going to vaccinate because it's too difficult to basically test everybody and figure out their status.
Mm-hmm. Thank you. Then another question is about the route of how the vaccine is delivered. And the question is, why are the RNA vaccines not applied via the subcutaneous or the intradermal route?
That works as well. So I'm actually not sure why the decision for those vaccines now was to do intramuscular. But in my lab, for example, also has experience with RNA vaccines. And we often do intradermal vaccination. It's pretty straightforward, and it works as well.
Mm-hmm. Michael, I don't know, would you like to go on with, because I think you also received questions?
Yes, there was also a question regarding will the measurement of neutralizing antibodies help to decide maybe if booster shots are needed?
Yes, in the long run, I believe that there's very good correlation between ELISA titers and neutralizing antibody titers. And since doing neutralization is a pain and you have to go to biosafety level 3, it's more likely that an ELISA titer will be used for that. But yeah, I think so. I think we will have a correlate of protection that might then determine if you need a booster dose or not.
Okay, then I think we will go on. And the next question was about the FDA and the EMA and what they exactly do. So what's their job? And do they really have to, so to say, believe the data which they get from the producers, or how can they actually verify the data?
Yeah, so the FDA, for example, does their own analysis too, right? They want to have the data sets and they run their analysis, right? So in a way, this is a regulated process. You can basically audit each and every step of it. The FDA at any point in time could say, oh, I want to see the data from that pharmacy in that medical center, which vaccine was given. I want to see that record. I want to see that record. And they have their own statisticians who look into those records actually, right? And it would look very, very bad for any of these pharma companies if what they deliver in their analysis looks different from what the FDA finds. So the FDA does that. And what you have to keep in mind, I know a lot of people at the FDA actually, they're just sitting at the desk and they're looking at data that somebody else produced, right? They have usually, I don't know if 50-50, but they have a split. They actually have labs. They work on stuff. They do research. So they know a lot of stuff and they kind of know what to look for. So those are not some kind of administrative guy that, just look at the data package and say, yeah, it's fine. Those are people who actually know what they're doing.
Then the next question would be about actually what happened with the AstraZeneca vaccine. So they showed that there was a 62% efficiency in Brazil, but 90% in the UK, although only half the dose was given to the participants. So could you speculate on the reason why this is so?
Yeah, I can speculate, and there's two explanations. The first explanation is, that's what AstraZeneca put forth, is that if you give the viral vector twice, you always have to deal with vector immunity, right? When you give it the first time, you might develop antibodies that then when you give the vector the second time might neutralize the vector, right? That's a pretty big problem with viral vectors. And they say that, because it was just half the dose, they didn't get a good anti-vector response, and that's why it worked better when they gave it the second time. The other explanation, which I think is simpler, is that there are so few people in that phase 3 in the UK that the confidence intervals are probably so wide. The result might be significantly different from the 62%. And if they get more data, it might actually approach the 62%. So those are the possible explanations, and we'll see. But I mean, a mistake like that is a real PR disaster. I mean, that's not, if you're a big company like that, it's just not supposed to happen.
Yeah. It doesn't give vaccine development a very good name, maybe in this case, yeah.
I mean, you just to respond to that, you can see it from a different angle too, right? Mistakes might happen, but they find them and they have to publicly communicate them. They didn't hide it.
That's true, yes. It's good to look at it that way, that's true. So we have another question. This is also a little bit speculative maybe, and about your own opinion probably. What is your estimation on when the pandemic will be over? Now that we have 2 really good vaccine candidates and also others coming up.
All right, I really can only speculate about that, okay? Don't tell me in half a year I was wrong, it's just speculation. There's a couple of things that come together here. Historically, pandemics usually take 1.5 to 2 years. If you look at the last 3 flu pandemics, 57, 68, and 2009. And then we have vaccines that will be hopefully used quite a bit in the next few months, right? And then the last point is that, and we've seen that this year also in spring, the temperature gets warmer, the virus has a harder spread. And so these three factors, in a way, or two of these factors, the vaccine and the rising temperatures, come together and might bring the situation back to almost normal in late spring. And then I think if you use the summer well and vaccinate a lot of people, I think in fall this might not be a big problem anymore.
There was another question about actually the immune response. So is there any correlation between the strength of the innate immune reactions following the injection and the subsequent antibody response?
You would speculate that that is the case. I don't know for this vaccine trial if somebody has looked at it specifically. What is remarkable is that the antibody response is pretty uniform, right? I showed you that the antibody response of the natural infection varies quite a bit. That's not happening with the vaccine-induced one. And so if only the people who make a innate immune response that gives them reactogenicity react well while the other ones don't, that's not what we really see. But I don't think anybody has done a good analysis of that data set yet.
And maybe also additional to this question, are you concerned about the potential immunological cross-reactivity of the mRNA vaccines?
To what?
So from the immune system, can the co-expression of the spike protein with own cells proteins break immunotolerance, or are there any potential autoimmune diseases which might arise? Or
Yeah, no, that comes always up, but I'm not concerned about that for a certain reason. If you get infection, the virus forces your cells to make tons of viral proteins, many cells. For SARS-CoV-2, mostly in the respiratory tract, but in many cases also in the gut, in your heart, in your brain, and an immune response primarily against the proteins in these cells and not against the virus particles, right? And so, there are some reports that severe infections can induce autoantibodies as well. But if that would really be an issue, we would be dealing with a lot of people that have now autoimmune disease after virus infection, and that's not the case. So I'm not worried about that. And the inflammatory environment, when you vaccinate and the cells express that spike versus half of your respiratory tract is infected, is also quite different. So I'm not very worried about that.
Mm-hmm. Then we have another question also on the vaccination, of course. Is there any data about vaccinations in pregnant women, infants, or children? And you think would vaccination be advisable for these people?
Very good points. Very heated discussions. Okay, let's start with pregnant women. There were, of course, a few pregnant women in the Pfizer trial. You were not supposed to get pregnant, but somebody always gets pregnant in these trials. But there was very limited data sets. The FDA did discuss that last week, and there's another panel that advises them, a CDC panel, and they also gave recommendations. And the recommendation was basically that people in risk groups, for example, healthcare workers that are pregnant, should not be excluded from vaccination, but they should talk to their physicians and then decide if they want to get vaccinated. That's what we know for pregnant women. Trials in pregnant women are getting started, so there will be additional data for that soon. In terms of children, with the high reactogenicity, I don't think that these vaccines will get licensed for children. You can't do that with the data that we have right now because there were no children included in these trials. And now they are doing HD escalation studies to go first in basically adolescents and then down to children. And based on the data, there will be a license or not. Right now in the US, the vaccine is licensed from 16 and upwards. And that was actually the most heated discussion point last week. If they should start at 18 or if it should start at 16. There was really a lot of discussion about that. So I don't think that these vaccines are going to be used in children very soon.
Thank you very much for that answer. Michael, do you still have questions that were sent to you?
Not specifically to me, but there was also a personal question maybe, but I might already know the answer following your talks. But there was, for example, a question that if you would have the possibility right now to get vaccinated, if you would do it. I mean, it's a frequently asked question to you, I think.
Yeah, but there's an easy answer to that. So I'm enrolled in the Pfizer trial. I'm part of the phase 3 study. So a lot of my colleagues at the department, and so is my wife. And I already recommended to my parents and grandparents that they should get vaccinated. So the question is not, would I take the vaccine? My main question now is the placebo control group or in the vaccine group, which I don't know yet.
There's another question concerning RNA vaccines in general. So is the liposome stability more an issue than the mRNA stability, or is both important?
The liposome composition is important in terms of delivery, also in terms of reactogenicity to a certain degree, and in terms of if the liposomes stay in the muscle or if they kind of basically disseminate. Yeah, it's mostly the RNA stability that is the issue. There are differences, small differences in the formulation between Pfizer and Moderna, and the Moderna vaccine seems to be slightly more stable. You can store it at minus 20 degrees Celsius instead of 60 to 80 for Pfizer, and it's longer stable at room temperature. Yeah, it's basically the RNA that is the problem.
Okay, thank you very much. I can go on in that case with another question that we have in the chat. First of all, almost all the people asking questions say thank you for the wonderful presentations. I think we should also add that to the questions. And yes, in the beginning, it was already mentioned that you have recent work on a universal influenza vaccine that elicits an immune response against the hemagglutinin stalk. So would a similar approach be feasible to develop a universal coronavirus vaccine that can help reduce the severity of potential future coronavirus pandemics?
Maybe. We're thinking, it's actually pretty funny. A few years ago, I started to talk to Matt Freeman at the University of Maryland. And he's a bona fide corona guy. He's working on MERS mostly. And we said, oh, start to look at epitopes that are cross-reactive between MERS and SARS and the human coronas. And we started, but it never got off the ground. And of course, we're now reviving that. And it might be possible. Who knows? It's hard. Because for flu, the stock is relatively conserved. The S2, which is the equivalent for the spike, the conservation is not that high. So it might be possible, but I think it's going to be hard.
Mm-hmm. Okay, then we have still many questions. Actually, we need to check to not also ask the same. Maybe we can go on with.
I think I will just compile more questions. So it's always about how stable the mRNA is and how long it will be in the tissue, or is it possible to be a systemic RNA expulsion, or what is your thought about that?
So in terms of how stable that is, how long it stays in the tissue and how long you get antigen expression, there's actually very, very good data. There's tons of mouse studies because you can actually package RNA for luciferase, for example, into these LNPs and then deliver them. And then you can image, right? You can actually look how long the fluorescence is there. And so it's just for a few days. There's basically the RNA is there for a few days, the protein expression is there for a few days, and then it's gone.
Mm-hmm. I will go on with this question, if you don't mind. A bit more of a technical, maybe even economical question, too, actually. So how do the production costs of different vaccine technologies compare? And is there a major difference between, for example, the novel mRNA-based systems to other systems? And also, as a second part to this question, your opinion is asked about whether the recent success of mRNA-based vaccines could make some other vaccine technologies obsolete?
Okay, the first. Hypothetically, I think making mRNA vaccines is easier and cheaper than making, for example, a vectored vaccine, just because it's more defined. It's a biochemical process, not a process where you need living cells and a lot of downstream purification. That doesn't mean that these vaccines are going to be cheaper. The price is a combination of many different things. It's a combination of how well a country negotiates with a company. It's the development cost goes into that. That might include how much money these companies took from governments. Pfizer didn't take any. Moderna was heavily financed by the Trump administration. So there's a lot of factors there. The second question, I think these RNA vaccines the platform is really interesting. We are evaluating it for many different viruses right now, but it's not magic. It will not replace a lot of the vaccines that we already have, just because then you would have to develop that vaccine as RNA vaccine, which would cost a lot of money. It might just replace something that is as good already. And you know, there are some things that are harder to tackle, right? Tuberculosis, universal flu vaccines, HIV, malaria, and you know, the mRNA vaccine technology might help to get that done a little bit quicker or a little bit better, but it's not like we use that now and we'll find a vaccine against everything. It's just not a magic bullet.
Unfortunately.
So I think we will have to go to get to an end, because also Professor Krammer can only stay till 6 p.m. So we will pick now a last question. So what are your expectations regarding long-term adverse effects of the vaccines?
That's hard to say, right? So we have pretty good data in about 40,000 people with the RNA vaccines. And I'm putting together the people who got vaccinated in the Pfizer trial and in the Moderna trial, the ones actually the vaccine groups. And there was little in terms of adverse events that would worry anybody. Now, the problem is that we have, of course, for some vaccines, rare adverse events that happen. The problem is not the problem. That's actually good. They happen very rarely. And so you don't pick them in phase 3 trials because you don't have enough people. Examples are Guillain-Barré syndrome for flu. For example, reversion to wild type for the oral polio vaccines, things like that. They happen very rarely and you don't pick them up in phase 3 trials. The important thing to keep in mind is that these things happen relatively quickly after vaccination. There's basically no experience with severe adverse events and vaccination where you said, oh, after 5 years, now we know that this happens. These things happen days to months after the vaccine is given, usually much quicker. And that's also one of the reasons why the FDA wanted to have 2 months of safety follow-up and not 15 years of safety follow-up before they kind of considered an emergency use authorization. So really long-term problems are not something that we usually see with vaccines.
So I think Maria has one last question.
I think it should be the last or second to last question at least. You have previously also published a commentary in Med, which is a journal from the Cell Press, with the title Pandemic Vaccines, How Are We Going to Be Better Prepared Next Time? And in the abstract, you're writing that what we could maybe do to next time when a new corona or virus hits us, what we could do to have a vaccine within maybe 3 to 4 months. So this is probably a long topic to talk about, but are there some short statements that you have in mind, what we would have to do, how we would need to be prepared for the next, hopefully not next pandemic?
Yeah, sure. Basically, you can come up with a strategy where you do a lot of surveillance. So you know which viruses are out there and you do a lot of characterization. We do that partially for flu pretty well, but not for other viruses. And you can pick viruses from these virus families where you know the bad guys are and where you know that it's likely that those virus families produce new emerging viruses. And you can pick 50 to 100 of these and start vaccine development. Go into phase 1, phase 2, get long-term data, on immune responses, and then when a virus hits, you just look what's the closest, you do a replacement, basically a strain change, and you produce and go into phase 3, and you have time to develop correlates of protection, so you license that with a correlate of protection. That could be done within 3 months, maybe 4 months. The only problem to do that's going to cost a lot of money. That's a few $billion, which so far wasn't of interest. So nobody wanted to fund something like that. But as we're considering it, if you compare that to how much money was lost now and how many lives were lost. So I think from the vaccine side, we can do many things to be better prepared. Of course, you can say the same about antiviral drugs, about better structures react to pandemics and about non-pharmaceutical interventions. But I think we can do much better. You just need the political will and politicians who actually have the foresight to do that and don't just think about the next election cycle.
So thank you very much. I think we will close the session now. Thank you for the very interesting talk. Thank you to the participants for the questions. There are really a lot of questions. However, we couldn't read all of them because there were too many, but this means that your talk was very, very insightful and interesting. I would like to say that the YSA will be on the Christmas break over the holidays, but we are looking forward to you participating in our next webinar, which will be in January, about biophotonics and light in medicine. We already have 2 speakers. It's Jennifer Barton and Wolfgang Dexter who will be talking. And just stay tuned on our social media, our website, our Facebook and Instagram to stay up to date to the following events. And if you're interested in the YSA and would also like to organize such a webinar like the one today, just contact us. We are very happy to talk to you and get your insights. So this is from our side, I think.
Thank you very, very much, Florian Krammer, for joining us. Yeah, it's a pity we couldn't ask all the questions, but I think we will probably be sitting 2 more hours. But yeah, you gave us really a very good educational lecture, actually. And yeah, I feel very well informed now, I have to say. I hope also the attendees too. Thank you very, very much.
Thanks for the invitation.
Lovely evening and enjoy your holidays.
Thank you so much. Goodbye.
Oh.